Welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. Rachel Ross will present Neuroscience of Stress and Metabolism. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of over 180 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that the research funded by BBRF has had such great impact is because we do not limit ourselves to one illness or condition. Since 1987, the foundation has awarded more than $440 million to fund more than 6,400 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Rachel Ross. Dr. Ross is an assistant professor in the departments of psychiatry, neuroscience, and medicine at Albert Einstein College of Medicine, and part of the Psychiatric Research Institute of Montefiore and Einstein in the Bronx, New York. She was a 2022 Young Investigator. Our webinar will begin with Dr. Ross's presentation, which will then be followed by Q&A. To submit your questions, please use the Questions tab on the control panel on your screen. Feel free to submit your questions at any time. Following the presentation, I'll ask as many questions as possible in the time allotted. And now I'm pleased to introduce Dr. Ross. Rachel, the floor is yours.
Great. Thank you so much, Jeff. And thank you all for tuning in to hear me talk about how we think about eating disorders a little differently than most, how we consider them across the weight spectrum, and how we use animal models to investigate the integration of stress pathways and metabolic circuitry in the brain. I want to start with making sure we're all on the same page with the definition of stress. It's any emotional or physiological challenge. These can be external or perceived internally. And I think we can all relate to the feeling that this person is displaying, where we might be thinking about a looming deadline for which we have a ton of work to do, and a bunch of different people are trying to get our attention. What happens in this situation? We might get a headache, we might get nauseous, you might get hungry. It's really different for everyone, but what's interesting to me is that the body is what responds to that thought or that feeling. Just in case I triggered you to connect with this stressful experience, I want to take a minute to provide a strategy for managing stress with a deep breath and calm visuals before we dive into the pathways of the stress response system. And let me tell you, I just did this before starting this talk. This is called box breathing, and I'll lead us through 2 rounds. I'll ask you to breathe deeply into your abdomen, starting with filling your belly. 2, 3, 4. Hold it. 2, 3, 4. Let it out. 2, 3, 4. And hold it. 2, 3, 4. Breathe into your abdomen. 2, 3, 4. Hold. 2, 3, 4. Let it out. 2, 3, 4. And hold. 2, 3, 4. This is a useful technique in general when you're feeling overwhelmed by stress, providing some calming feedback through your autonomic nervous system, which I'll tell you more about before I connect it to the metabolic system, and explain how in my lab, we're trying to understand how these two systems interact within the brain and what that may mean for understanding eating disorders. There are two pathways activated by stress, no matter if the stressor is positive or negative. The autonomic nervous system, specifically the sympathetic nervous system, is the coordinator of what we think of as the fight or flight response. It has a counterpart called the parasympathetic system, which is actually what we used in the breathing exercise. Both of these systems use super fast electrical signals originating from multiple regions in the brain that project out through the nerves to the trunk of the body to organize everything the body might need to respond to a challenge. Some of these signals stimulate the adrenal gland to produce the hormone norepinephrine, which gets converted to epinephrine, and you may be familiar with the other name for this hormone as adrenaline because of its origin from the adrenals. Epinephrine coordinates the body's response to stress in response to sympathetic activation. You'll have dilated pupils to let more light in for improved visual scanning, relaxed lungs to improve oxygen exchange, increased heart rate to get that oxygen out to the body efficiently, and the hormones that ensure that even the faraway organs get the same message. At the same time, and originating from some of the overlapping brain regions, there is a slower neuroendocrine or hormone-based system called the hypothalamic pituitary adrenal system, or the HPA axis, that's responding to these signals as well. Neural signals from across the brain converge on cells in the hypothalamus, which releases a hormone, CRF, corticotropin releasing factor, that acts in the pituitary gland and leads to another hormone, adrenocorticotropin hormone, or ACTH, released into the blood. ACTH targets the adrenal glands, the same glands that produce norepinephrine, but it acts on different cells in those glands to produce cortisol, the major glucocorticoid hormone, which functions to similarly prepare the body for a sustained stress response, specifically nutrient release from organs that store them to maintain energy levels over time. Cortisol and norepinephrine both feed back to the brain and influence the dynamic connections between neurons in these stress-responsive networks, creating the opportunity for faster or more robust responses should a similar stressor reappear. In the short term, the coordination of these systems is a great thing, but in the long term, you might appreciate some complications of having an elevated stress response, which is what we commonly see in the symptoms of post-traumatic stress disorder or in the presentation of anxiety and depressive disorders. Most likely, the whole brain is involved in the coordinated response to stress, but the primary regions to consider due to their regulation of or response to the stress hormones are shown here within the brain. The prefrontal cortex is a region of the brain that's involved in general cognitive function, social and self-awareness, decision-making, and behavior inhibition, and we'll come back to this more in detail later. The amygdala is a region of the brain most well known for its involvement in fear response, though it's also likely important for any valence-based behavior response, whether positive or negative. The hippocampus is the seat of memory formation, and it, as well as the amygdala, are the most sensitive regions of the brain, inasmuch as both are highly responsive and plastic over time to outside changes. This could be the source of memory difficulty and hypervigilance that we see in the setting of chronic stress. On the other side, the hypothalamus and the pituitary, which happen to be buried in the center of the brain and therefore are not really shown in this picture. These are important drivers of the hormonal stress response, and they also respond to the circulating stress hormones through negative feedback, providing the opportunity, say like a smart heating system, that can reset in response to external and internal changes. These are the regions that are known to be activated in the setting of acute stress and are associated with the range of physiological, behavior, and emotional reactions to stress. Now, in mice, it's rather complicated to observe emotional changes in response to stress. However, mice give us access to so much more information than we can understand from humans in order to understand the brain. So I'll introduce you to the stress circuitry in the brains of mice, and then explain how we can use mouse models with good fidelity to appreciate the many levels of complexity that will provide better opportunity in time to improve our diagnostic clarity and treatment for complex stress-related disorders. So this is a schematic view of the mouse brain, which is smaller and a little simpler than the human brain, but has many of the same general regions. Here I've listed in purple a number of the regions that are involved in the stress response. In particular, I want to draw your attention to two hubs. This first hub is what we think of as the limbic system, the neural stress response system that relays information within the central nervous system. And here you can see it has the same regions as the human brain, including the medial prefrontal cortex, the hippocampus, the amygdala, and the hypothalamus. Here you can see the paraventricular hypothalamus and the lateral hypothalamus. These circuits together encode signals about the importance of particular information, previous experiences with that information, and whether the information carries a positive or a negative connotation. A second hub centers around the PVH, paraventricular nucleus of the hypothalamus, and this area initiates the HPA axis signal outward toward the body. It also plays a role in coordinating the sympathetic nervous system signal 2 with projections to the brainstem region near this area called the locus ceruleus, which happens to be dense with cells that produce the signals that we're generally familiar with in psychiatry, like serotonin and norepinephrine. But you may appreciate that our understanding of how these signals are playing a role in behavior and function remains poorly understood. That's really because the mechanisms are even more complex than we've been able to appreciate to date. In order to study these complex diseases that result from stress, we generally use a reductionist model, starting with this bird's eye view that I'm showing you of a park. We step down from there to achieve more detail and higher resolution, getting into the forest view, to the single tree, to the branches of the tree, to a leaf. My lab and others studying neuroscience applied to psychiatry focus on objective translatable behavior modules, like exploratory behavior or heart rate. And this gives us something objective and ethologically relevant to study in animal models that is also relevant and directly translatable to humans. This is very different than historical studies using animal models of genetic diseases that are only seen in humans or may be considered targeting a chemical imbalance. These behavior modules relate to the human experience across disease paradigms rather than directly connecting to one. And I'll go into that a little bit more when I talk about food intake. But in general, consider any behavioral observation as being a correlate to our view of the park. As we take it one step down into the forest, we have tools that let us investigate the regional circuits within the brain, which can be done in humans or in animals. And this is where many newer neuromodulatory therapies are focused. If we go deeper, we can resolve individual cell populations and measure their activity, which is something that's really only possible in animals and in the last 10 years. When we zoom into the branches and the connections, we can investigate the interactions between these cells, which could be chemical or electrical. And when we get all the way to the leaf components, we can consider individual proteins and the genes that define them, which is commonly where our medications are targeted. So I'm going to tell you how in my lab, we access this from both directions by studying a behavior, food intake, and a molecule, the melanocortin 4 receptor, to understand the circuitry and circuit-based interactions underlying normal animal behavior and the maladaptive behavior that can be associated with disorders of body weight in humans, ranging from anorexia to obesity. Now, eating disorders are a spectrum of ailments that involve dysfunctional feeding behavior and can also include altered body weight. We tend to have a distinct visual idea of what people with eating disorders look like, but clinical data shows us that bias serves us wrong. Across the board, eating disorders affect 9% of the US population, and most of these people are not actually underweight. I see this quite commonly where I work in the Bronx. Binge eating disorder actually has the highest prevalence of all eating disorders in the population, and people with this disorder are commonly overweight. On the other side of the spectrum, anorexia nervosa affects nearly 1 to 3% of the US population, is more common in people assigned female at birth, and has the highest mortality of any psychiatric disorder. While the majority of people who suffer from anorexia do experience remission over time, the longer a person suffers the disorder, the more significant stress on the body, and the less likely they are to remit. Eating disorders are highly heritable, and genes associated to metabolism and neuroprotection are most commonly associated with illness. Anorexia specifically has been the subject of a recent large-scale genetic studies called genome-wide association studies, which have shown multi-trait associations with findings that link to body mass index, type 2 diabetes, and cholesterol, indicating underlying risk similarities to disorders of metabolism that are associated with being overweight. These GWAS studies have also linked the risk factors for anorexia as overlapping with those for obsessive-compulsive disorder, major depressive disorder, anxiety disorders, and schizophrenia, suggesting more similarities than differences across the stress response system as well as the weight spectrum. In restrictive eating disorders, no matter what the body weight, people choose to not eat even though they're effectively starving. And the experience of starvation is an obvious and significant stressor that leads to all sorts of body responses. For some people with restrictive eating disorders, the preference to not eat may be an attempt to mitigate some other stressor that's causing discomfort. In effect, self-starvation can be a way to tamp down a stress response that would otherwise induce less appropriate behavior, which is something I often see in my patients with comorbid OCD or social anxiety, along with anorexia. When people are able to maintain the starved state for an extended period of time, this becomes an additional stressor that's harmful to the body. Over time, this leads to all the symptoms that we associate with anorexia clinically: cognitive impairment, anemia and poor cardiac function, weak muscles and bones, and difficulty with digestive, reproductive, immunologic, and endocrine function. An interesting thing about anorexia, unlike every other psychiatric disorder, is that the presentation forces upon us the recognition that the brain and the body are connected. All of the effective treatments, whether psychological or medical, start with restoring body weight. In psychiatry, as well as in neuroscience, we tend to forget that the brain is connected to a body, and that the things that come from the body, like circulating hormones or signals, provide information such as nutrient status and availability that effectively coordinates the internal milieu in which the brain is working. This includes the stress-related hormones that I mentioned previously that are important in psychiatric illness presentation, as well as the metabolic hormones listed here that I'll go into next. But again, in most eating disorders, even binge eating disorder, no matter what a person's body mass presentation, they will spend a significant amount of time restricting food intake, which creates a chronic stress on the body and the brain, which has the potential to create dysfunctional coordination in these overlapping stress response and metabolic systems. So I'll pause here for take-home point 1. Eating disorders are a spectrum of illnesses where the capacity to eat, unrelated to metabolic need, can result in both larger and smaller bodied outcomes. The way that we've categorized eating disorders to this point has led to a lot of stigma and confusion for translating to biology, diagnosis, and to treatment. Since most of psychiatric illness involves some upsetting of the stress response, and since eating disorders themselves create a significant stress on the body, thinking about the weight spectrum suggests that it may behoove us to pay attention to the regions that interface between the stress and the metabolic systems. But before we can go there, I would like to introduce you to another set of hormones, the circuitry, and also the cells and proteins that regulate metabolism, which are actually quite similar and in some cases overlapping with those that regulate the response to stress. Here I'm showing you a simplified representation of the opposing body pathways involved in regulating the metabolic system. Unlike the stress response system, these work opposite each other rather than together. In a well-nourished, healthy state, fat levels are replete and produce the hormone leptin, which signals through the body that there is energy available to spare. When sugar and carbohydrate are available, like after most of our Western diet meals, the healthy pancreas secretes insulin, which directs energy storage in the body, and along with leptin, provides signals to the brain that help it maintain normal function and also tell it that it can stop eating. In the normal hunger state, as well as in the starved state, the empty stomach secretes the hormone ghrelin, which opposes satiety hormones and encourages the body to conserve energy and signals to the brain that to motivate food finding and eating. Recall that these are some of the regions involved in stress response that I'm showing in purple. There is some overlap with the regions that are involved in metabolism, now I'm showing in pink, particularly within the hypothalamus. Again, in this system, there are two main hubs for metabolic information, and most have dual direction communication, quite like the stress response system. The brainstem is home to multiple regions. Here I'm showing the nucleus of the solitary tract and the parabrachial nucleus that are directly responsive to the metabolic hormones, as well as to the neural signals that come from the gut in response to the nutrients of food. And these relay with the paraventricular nucleus of the hypothalamus, as well as with the lateral hypothalamus, both of which are also integral to the stress response pathways. These regions, as well as the arcuate nucleus at the base of the hypothalamus, and a few more that I've left out for simplicity, are the original feeding and satiety centers of the brain, known since experiments done back in the 1950s to be integral to survival and the drive to eat. If we zoom in more deeply within the arcuate, we can see that the hormones that correspond to nutrient need, like ghrelin, and nutrient abundance, like leptin and insulin, can transmit their unique signal through a leaky blood-brain barrier in the arcuate nucleus of the hypothalamus, which activates two populations of neurons differently. These two populations produce proteins, or peptides, that both act on the same downstream receptors, called melanocortin receptors, but have opposite effects. The canonical feeding neurons, which produce the peptide AGRP, are activated by ghrelin, but inhibited by leptin, and act downstream on the melanocortin receptors, leading to the outcome of food intake and energy conservation, consistent with the feeling of hunger. The POMC neurons, which are found right next to the AGRP neurons in the arcuate nucleus, are activated by the hormones of nutrient availability and release a protein called alpha-MSH, which has the opposite effect on the melanocortin receptors, stimulating them and reducing food intake, as well as increasing energy expenditure, as would be possible in satiety. And one of the things that I find so cool about this system is that the hormones have opposing effects on the neurons, and then the neuropeptides produced by these neurons have opposing effects on the melanocortin receptor, and this working together leads to a coordinated but opposite behavioral response. In terms of brain systems, few of which are binary in this way, it's actually relatively simple if you can keep the players straight, which I will try to do throughout the remainder of this talk and help you through. Now, the melanocortin system is a set of 5 receptors that have multiple endogenous ligands. I'm going to focus on one of those receptors, the 4th melanocortin receptor. It is primarily expressed in the brain. In humans, the melanocortin 4 receptor gene is the most common single gene linked to obesity, and there are many mutations within the gene that make it non-functional that are associated to obesity in humans. But in mice, as well as we see in humans, global deletion of the melanocortin 4 receptor leads to massive obesity. Animals are approximately double the size of their wild-type counterparts. And this is quite apparent to us to see in mice, which is quite different from their emotions. This is a schematic of the important regions of the brain where melanocortin 4 receptors are known to be expressed, though general investigation has been limited to the parts that are known to be involved in metabolism. All of these yellow circles are brain regions where melanocortin 4 receptor is found and are involved in the regulation of food intake and energy metabolism. The arrows that lead to them are projections from the neurons at the base of the brain, the arcuate nucleus of the hypothalamus that I just talked about, that produce the proteins that either stimulate or inhibit the melanocortin 4 receptors directly. The blue arrows represent projections from the feeding neurons, AGRP, which are found only in the arcuate, and the red arrows represent projections from the satiety neurons, called POMC, which can originate from both the arcuate and the brainstem. Recall that AGRP inhibits the melanocortin 4 receptor to stimulate food intake, and alpha-MSH from POMC stimulates the melanocortin 4 receptor to inhibit food intake. These both also have effects on energy expenditure through the body, which are very important for body weight management and mounting an appropriate response to stress. But since we're talking about eating disorders specifically, I'm going to focus on the impact on food intake. The capacity to eat when not hungry, or to not eat even when hungry, is a remarkable feat that animals will not perform unless they're perturbed in some way. For a number of years, researchers focused on the metabolic hormones, looking for biomarkers or dysregulated responses to the starvation state that might explain how an animal would eat or not eat outside of its need state, as we see in eating disorders. But the metabolic hormones are generally responding as expected to nutrient state, whether nutrients are scarce or seemingly abundant. In anorexia in particular, a major biomarker is the impact of starvation on cognitive function. People with anorexia have reduced cognitive function and difficulty with flexible thinking when they're in the restricted state, much of which is reversible once they return to their normal weight. However, as I mentioned, much of the research on anorexia, particularly in animal models, has focused on the metabolic impairment and otherwise neglected this cognitive dysfunction. So this is where we'll get into the data from my lab, because when we started this work, no one had investigated the cortical regions like the medial prefrontal cortex, that region of the brain in mice and in humans that's responsible for cognitive function and involved in stress response. To study this, we used mice that have been engineered to express a green fluorescent protein in the cells in which melanocortin 4 receptor is also expressed. We take a slice through the medial prefrontal cortex, which you can see here shows that there's quite a lot of green and therefore a lot of melanocortin 4 receptor throughout these cortical regions. The box delineates the ventromedial prefrontal cortex region, which in the mouse has specific subregions that we've labeled. Each of these subregions has slightly different roles in regulating cognitive function. The dorsal region, which is more shallow in the brain, is the prelimbic area, and the deeper region is the infralimbic area, and deeper still are the dorsal pedunculum and the dorsal tenea tecta. In each of them, there seem to be a large number of cells that express melanocortin 4 receptor, and for the remainder of the talk, I'll be referring primarily to these two regions, the prelimbic and the infralimbic. So a brief pause for take-home point 2. I've shown you that the metabolic system is similar in organization to the stress response system, and one of the major players, the melanocortin 4 receptor, is found to be highly expressed not just within the brain regions that are important to metabolism, but also in at least one region, the medial prefrontal cortex, that is important in coordinating stress response through its role coordinating cognitive function. There have been very few mechanistic studies in animal models looking at metabolic-related circuitry and cognitive function, though there is some evidence in human studies that this is important across the weight spectrum. So I think it's important to look at this region of the brain, which isn't directly involved in body weight regulation, but it could provide us new insight and treatment opportunities for eating disorders across the weight spectrum. Cognitive function in humans includes planning complex behavior, organizing potentially conflicting inputs and outcomes, and engaging self-control in social settings, and coordinating the activity that results from this integration, all of which together can be described as executive function. These functions are organized by the extended prefrontal cortex, shown here in green, yellow, and red in a schematic of the human brain. As you may recall, this area of the brain, as well as the others shown here, are known to be involved in stress response, but unlike some of these other areas, the prefrontal cortex has not been directly connected to metabolism and metabolic circuitry. To our knowledge, rodents have a less complex prefrontal cortex region, corresponding in this diagram to this region, the ventromedial prefrontal cortex, as well as parts of the anterior cingulate cortex here in red. These are the infralimbic and prelimbic regions that I just introduced within the mouse brain. Activity in these regions allows mice to perform simple executive functions, such as cognitive flexibility, the ability to change an association based on new information, input organization, the ability to take two conflicting pieces of information and choose an action, and behavior inhibition, the ability to hold back from engaging in a particular action. These three domains of cognitive function actually overlay well with the regions that show functional MRI activity changes in body weight related studies in humans and functional difficulty in neuropsych testing in people of different body weights. In this radar chart from an analysis done about 10 years ago, you can see that the domains of cognitive function are differentially impaired across the weight spectrum. Here, the red line represents people with obesity, green is the standard weight control, and blue is people with anorexia. You can see that decision-making function is reduced both in people with anorexia and people with obesity compared to the standard weight controls, and cognitive rigidity is enhanced, or you can consider this flexibility is reduced in both people with obesity and people with anorexia. People with obesity seem to have reduced capacity for interference control compared to the standard weight controls, and people with anorexia seem to show enhanced control. These data have been recapitulated in recent human imaging studies, which I'll talk about briefly here. In one, a group brought people with different body habitus into the lab, underfed them prior to showing them images of high-calorie food, so they're hungry, and then they obtained a functional MRI of the whole brain, and in this they showed that the obesity-prone folks, compared to normal weight or obesity-resistant individuals, have increased activation in multiple regions of the cortex, including the medial prefrontal cortex, in response to images of energy-dense or rewarding food like these fries. This is interesting to us since we found in our animals that the melanocortin 4 receptor is expressed throughout these regions as well. In a study focusing on the ventromedial prefrontal cortex region in particular in humans, people across the weight spectrum were asked to choose between healthy and unhealthy food options. And in this study, the researchers saw an inverse relationship of activation in the ventromedial prefrontal cortex region that varies with hunger state and is inversely related to weight, suggesting to me that the hunger-related information must be carried to this region of the brain to provide internal context in the function of decision-making. Finally, this region of the brain actually is responsive to hunger state. Here you can see brain activity measured by fMRI. This bright spot shows higher activity in study subjects in different nutrient states in response to these types of images, different images of food. The graph here quantifies the activity and shows that in people who are hungry, this white bar, the activity levels are increased when they see delicious or high-fat food, but not when the people are full, here in gray. So we have this particular region of the brain that's specifically sensitive to energy state, and over time, this sensitivity could lead to alterations in behavior similar to those that we see in the setting of chronic stress. In fact, if you happen to watch the October series of the Meet the Scientist series, Sahib Khalsa spoke about eating disorders and showed from their lab when they provided a drug that mimics the effect of the stress hormone epinephrine released acutely before a meal, they saw increased activation of this ventromedial prefrontal cortex region in people with anorexia, more so than in healthy weight controls. So in these people with restrictive eating disorders, the stress input in the hunger state has a greater impact in this region than would be experienced by those people who do not have an eating disorder. And this interaction may be important because not eating for extended periods or being hungry is quite aversive in nature. I think that most can relate to this through the lived experience of hunger, but it's also been shown in mouse studies. So I'll tell you about a study where they stimulated the melanocortin neurons of the PVH, which, if you recall, is equivalent to providing input that food is available and reduces the feeling of hunger. They used a behavior task called real-time place preference, where they put animals into a two-sided chamber, where one side provides the opportunity to stimulate this satiety circuit, and the other side does not. They allowed the animals to choose whether or not to experience the stimulation in both the hungry and the sated state. The white bars here represent control state with no stimulation on either side of the box, but the animals can still choose where they spend their time. And here you can see they have no preference for either side of the box. But in the situation where stimulation is available on only one side of the box, you can see the animals increase the amount of time that they spend on that side in only the hungry state. In the sated state, the stimulation presents no preference bias. If we extrapolate this to appreciate how it may play out in people, we might consider that the choice to not eat, which is aversive at baseline even for those who maintain it, may be a way to reduce other unpleasant feelings. And these may come from other stressors, as I see in my patients. But to date, we don't really know how hunger state information may reach the medial prefrontal cortex. And that gets us back to data from my lab. Recall that we really have to use animal models for this type of work in order to get the opportunity to visualize the circuitry on a molecular level, here with the trees and the branches. And you can see in this close-up image of a slice of a brain from the infralimbic region of a mouse that has green fluorescent protein marking cells that express melanocortin 4 receptor. This is the same brain regions that I showed you before. If we zoom in a little bit closer on that region, you can see the projections from AGRP neurons, these yellow dots in a string right here, and on POMC neurons, these pink dots kind of again on a string. These are projections from AGRP and POMC neurons that line up right near the green melanocortin 4 receptor expressing neurons, indicating that the neurons in this region get inputs from the hypothalamic feeding and satiety neurons both. So take home point 3. Putting this all together, in humans, we see the involvement of the medial prefrontal cortex region in response to hunger. The functions that it regulates are less robust in weight-related illness and can be impacted by stress, which may be due to hunger state information carried by melanocortinergic circuitry, which is something that we have discovered between the hypothalamus and the infralimbic region of the medial prefrontal cortex. Recall that there are multiple regions within the brain that are involved in both the stress circuitry, mostly the purple regions here, and the metabolic circuitry, the pink regions, and these stippled regions are involved in both. The stress response regions, the amygdala and the lateral hypothalamus, have been shown to participate in the integration of motivated drives, which might help us understand better both how the brain works as a computational system and how it's involved in both metabolic and psychiatric illness. I recommend checking out the work from Lori Zeltzer, Mark Anderman, Carrie Ferrario, Anne Graybiel, Scott Kanowski, Mike Crashes, Gorica Petrovich, and Scott Sternsen's groups for some great investigations within the hypothalamus, reward circuitry, and fear circuitry to better understand this perspective on conflicting motivational drives that may affect eating behaviors and relate to eating disorders. My lab is investigating this novel circuit from the arcuate to the medial prefrontal cortex to appreciate how executive function may integrate all of these inputs to regulate the balance between healthy and maladaptive behavior. But first, I'll highlight the work of a couple of other groups that have very recent papers also looking at this interface between metabolic circuitry and stress response circuitry, but in the opposite direction. In particular, they've looked at the projections from the medial prefrontal cortex to the lateral hypothalamus, a region of the brain that's critical to motivated behavior across various survival-based needs. In a very recent paper, a group in China has shown that input from the anterior cingulate cortex in mice, a region whose function is reasonably equivalent to the ventromedial prefrontal cortex in humans and also involved in cognitive function, sends projections to the lateral hypothalamus, which then projects to the arcuate nucleus of the hypothalamus. And they showed that this circuit changes feeding behavior. They do this by inducing the expression of a novel receptor in just this circuit, and then use an exogenous drug to inhibit that receptor and thereby block the signals of the circuit. This is called chemogenetics. They put this in the context of pain, as animals and humans experiencing significant pain stress tend to decrease their food intake. They used a behavior test for real-time place preference, where they put the animal in a two-sided chamber, but in this case, one side has food and one side does not. And they measure the outcome of the amount of time the animal spends in the food zone as an indirect readout of their interest in the food, and they also measure the amount eaten. As you can see in their data, this top heat map shows the animal in pain spends its time in the no food zone, and does not eat much food, depicted as the blue bar in the graph. But the animal in pain that receives inhibition of this cortical to hypothalamic circuit actually does increase its time spent in the food zone, as well as the amount that it eats during the trial, which is depicted by the red bar in the graph. This shows that manipulating this regionally defined circuit can overcome stress-induced reduction in food intake. In an even more recent paper, a group in Australia used a different mouse model to test the molecular identity of a subject of the subset of projections from the medial prefrontal cortex to the lateral hypothalamus that is activated by acute stress. And when they stimulated this circuit using chemogenetics, they were able to suppress motivated food intake, shown here as a decrease in the amount of foods the animals eat after being fasted overnight. In this case, the blue lines represent animals in who the circuit has been stimulated. But they also inhibited the circuit, and when they did this, they were able to alleviate the stress-induced suppression of food intake. Here, the blue bar represents the animals in whom the medial prefrontal cortex to lateral hypothalamus circuit has been inhibited, and they investigated the way an animal interacts with food in an aversive, novel environment when they're in the hunger state. In the animals who have this circuit inhibited, they start eating faster and they eat more overall in the aversive setting compared to animals who do not have this circuit stimulated. So these groups' work have provided evidence that the regulation of specific circuits from the medial prefrontal cortex impacts food intake, which reduces food intake in the setting of stress. And this could be relevant for restrictive eating disorders. It may even be overactive in some people with anorexia nervosa. And there's a chance that the neurons that they describe in the medial prefrontal cortex may be neurons that we have seen express melanocortin 4 receptor, which I hope we can investigate in the future. But first, I want to show you the work from our lab that shows the opposite direction of information relay from the arcuate hypothalamus to the medial prefrontal cortex that also impacts food intake and may provide the information that the medial prefrontal cortex is using to compute which motivated drive to follow between stress and metabolism. As I mentioned, in basic neuroscience research over the past 10 to 15 years, we've had the privilege to use some amazing new tools that let us basically turn on and turn off cells and circuits in a time-based way using remote control access, which, as far as we can tell, doesn't perturb the mouse too much. And in the work I just showed you, they use drugs, but in the work in my lab, we're using light, which is a process called optogenetics. And we use these optogenetic techniques in the awake and functioning mouse to manipulate our novel circuit of melanocortinergic projections. Specifically here, we stimulate the ends of the projections from AGRP feeding neurons within the medial prefrontal cortex only. And here we see that when the light is on, the mice eat significantly more over the course of an hour compared to the baseline when their projections are not stimulated. And this is really exciting because it indicates that these projections that we've seen are actually part of a functional circuit from the hypothalamus to the medial prefrontal cortex that can coordinate food intake directly. We're currently in the process of doing the same experiments with the POMC neuron population to see if we might appreciate the opposite findings that would be expected from stimulating the neurons that stimulate the melanocortin 4 receptor and may induce cessation of feeding. So stay tuned for that in the future. In order to understand the molecular level of what is this receptor doing here, we deleted the melanocortin 4 receptor from the infralimbic region in adult male mice, which allows us to investigate how the melanocortin peptide specific circuit is important in this region. We focused on the infralimbic region specifically because that's where we saw the projections coming from the feeding and satiety neurons. So this is where we expect the circuit will have a most direct impact. We are also studying this in female mice, but we keep our cohorts separate and we analyze the data separately since we expect there will be sex differences between the outcomes. So there will be more on that in the future as well. But when we deleted melanocortin 4 receptor from both sides of the mouse infralimbic region, these animals showed increased body weight, approximately 20% change in body weight compared to where they started before having the surgery. And they also show increased food intake, here grams over the course of 24 hours, Both of these are significant and tell us that the peptide signal that changes with food intake with our optogenetic stimulation likely requires the melanocortin 4 receptor. Compared to the whole body knockout, the image I showed you of the very large mouse, where the animals are nearly double in size with their wild type counterparts, this may not seem that remarkable, but this is really the only other region outside of the hypothalamus where manipulation of the melanocortin 4 receptor directly impacts body weight through food intake. And if you think about a change of 20% of your own body weight, whether it's up or down, you might appreciate that that's a really significant amount. So we also took advantage of the difficulty of these types of experiments where we are putting proteins or doing tiny infusions into these very small areas of the brain. And we analyzed the data from animals in whom we weren't quite able to hit our mark, but we hit regions that were around it or included it as well. And in this case, we looked at the animals from whom we deleted melanocortin 4 receptor from the prelimbic region, that more shallow part of the medial prefrontal cortex, or as well as the infralimbic region together, since this prelimbic region is known to be involved in decision-making and stress response as well. And in these animals, we see the opposite effect. The animals gain far less weight compared to the control animals that have had sham surgery, so no injection. And this shows that the same receptor in different regions of the brain can have opposing effects on weight regulation, and reiterates why this type of investigation, with an awareness of both the spatial and molecular identity of neuronal subpopulations, is crucial to building a full understanding of the brain. So my fourth take-home point. It seems that melanocortin 4 receptor in the ventromedial prefrontal cortex is bidirectionally involved in body weight regulation, which underscores the importance of thinking across the whole weight spectrum, and also the importance of using animal models to understand these larger brain networks, which is very relevant to any potential utility of the treatments for eating disorders, as well as other psychiatric ailments. In the last few slides, I'll show some of our work investigating how melanocortin 4 receptor in the broader ventromedial prefrontal cortex is or is not involved in elements of executive function directly or decision-making related to food. We've used a couple of complex behavior tasks in order to assay specifically for the domains of cognitive function that have been associated to body weight. First, to consider interference control, specifically choosing to eat in an aversive environment. We looked at the animal's behavior in the home cage after a fast in order to get their baseline, and we quantified their interaction with a pellet of food. You can see here that there's no difference between the control groups and the knockout groups in terms of the time they take to approach the food or to begin to consume the food in the home cage. However, when we put these animals into a novel context, which is an aversive experience, and we put a food pellet into the center of that novel context, we've provided a conflict of input. The animals have to manage this in order to choose their action response. Will they eat the food in the center? It's an appetitive signal, but being in the center of the field is aversive to them. So here you can see the knockout animals take a longer time to both approach and to consume the food in the setting of this conflicting input, which indicates a deficit in behavioral inhibition or the interference control element of executive function, the same domain in which people with obesity and people with anorexia have differing outcomes. Our melanocortin 4 receptor knockout mice are obese, and they show impairment in this function, much like people with obesity. Here, the animals are overvaluing the aversive input or taking longer to integrate the conflicting inputs. We also used an appetitive reversal learning paradigm that allows us to test cognitive flexibility. And here we set up a task that uses odors as cues and digging as a learned action in order to access the food. We trained the mice in a calorie-restricted state so they could learn to associate a particular smell with access to the food reward while ignoring a second smell until we switch the association so that they need to unlearn the original association and now attend to a previously unimportant second cue. This provides us an opportunity to study learning, a cortical function that's not related to metabolism, but often is implicated in the assumptions we make related to body weight. Here we saw that deletion of the melanocortin 4 receptor from the medial prefrontal cortex has no effect on the animal's capacity to learn the association of the first odor to the availability of food. There's no difference between the knockout and the control animals. And when we test their ability to update the association, we see again deletion of the medial prefrontal cortex from the melanocortin 4 receptor from the medial prefrontal cortex has no significant effect on perseveration. The animals are able to update this association. So we think that melanocortin 4 receptor in this region is actually not relevant to cognitive flexibility in the chronically food restricted state. And in fact, other groups have shown that this behavior may be regulated by a different region of the cortex in mice. Last take-home message. Altogether, I've showed you that melanocortin 4 receptor in the medial prefrontal cortex is critical to body weight maintenance through food intake. This seems to also play a role in unique executive function domains, such as decision-making related to food when conflicting inputs are presented at the same time, but not when the animal needs to update learned information that's associated with food. Since we know executive function is affected by body weight and impaired by stress, in the future, we hope to look at how these cells that express melanocortin 4 receptor in the infralimbic region and the related circuitry coming from the hypothalamus might be impacted in different hunger states and in response to stress. We think that interrogating the circuitry in mouse models across the weight spectrum while investigating cognitive behavior relevant to food intake will provide us with new appreciation for how eating disorders develop in humans and new targets for treatment that might help people who suffer from them. In fact, the funding we received this past year from the Brain Behavior Research Foundation will allow us to do exactly that. We are using these systems neuroscience tools in awake-behaving mice to see how inputs from the stress pathways, such as those from the amygdala, may interact with melanocortinergic signals coming from the arcuate nucleus and drive the behavior that we and others have seen to reduce food intake, possibly through inputs to the other motivation systems in the hypothalamus. I want to thank you so much for your attention, and a big thank you to my group and to our collaborators at my current and former institutions who have enabled this work, to my patients who inspired it, and to the institutions, acknowledged as well as those that are not, who support it. I want to specifically thank the Brain Behavior Research Foundation for being one of the only funding agencies to support research on eating disorders. I'm happy to take any questions.
Rachel, thank you for an excellent presentation and for the work that you're doing. Very exciting, and really, I think the presentation shows the connection between very basic work and what could ultimately come along clinically from that. So thank you for that. I want to ask you, you spoke about a few specific ongoing work that you're doing, and we're going to have you come back to share that with us. I'd like you to tell us, where do you see this going in the next 5 or 10 years? What can people look forward to in terms of they themselves or their loved one who may have an eating disorder?
In the next 5 to 10 years. Well, so again, because I tend to think of eating disorders across the weight spectrum, thinking outside of psychiatrically associated eating disorders, there has been some recent drugs that have come out that have effects directly targeting the metabolic system that actually seem to help people reduce their drive to eat. So this can be really helpful for people with binge eating disorder and people who have overeating behavior that can lead to obesity. And so I think the nature of those, considering those targets, and also thinking broadly about how those targets might impact the stress response and the motivation systems, I think there is promise that we might actually have some medications coming soon. I don't know if I can say within 5 years, but maybe on the 10-year frame that would help people who are on the under-eating side as well. I mentioned some work that's being done, I think, looking at the way these different systems interact on a circuit level. So not about drug targets per se, but really about those neuromodulatory therapies. And I think we are getting pretty close, that might be in the next 5 years, to consider some of those, they're little different types of therapies like TMS or DBS. I wouldn't go to DBS yet, but maybe TBS, where we stimulate different regions of the brain. And that might impact people's sort of habit-based circuitry that keeps them rigidly restricting their food intake. And in doing that, we might open up some opportunity to think differently about, or for them to think differently about how they engage with food. And I could see that being some new useful treatments. And I know that some of that with DBS actually is currently in clinical trial right now.
Great overview, and I think that we've seen how TMS is potentially helpful for a number of conditions and maybe for some of the eating disorders as well. One last question for somebody listening right now who they themselves or a loved one has an eating disorder. What should they do now? What's the best approach for somebody to get help for this situation?
Sure, great question. So I think first, if you have a family member who you think is suffering with an eating disorder, start by listening to them and being supportive. It can create a lot of conflict, I think, where we sort of blame people for their difficulties, and that gets in the way of accessing treatment, especially with eating disorders, where, and I'm thinking of anorexia in particular, but people generally with anorexia want to keep the anorexia, which is not necessarily the case with other eating disorders. So starting with being supportive, looking to see what resources are available in your community, and there's very wide arrays of resources that are differently available. There are some online resources, National Eating Disorder Association. In different states, there's state-based eating disorder associations that provide help with accessing clinicians with specialties that allow them to work with people with eating disorders. And then looking within your general healthcare network, wherever you are, but making sure to ask for people who have that specific expertise, because it does require having knowledge of medical body endocrine function while also thinking about what the person is presenting with. Some people, depending on the severity of the eating disorder, might need hospitalization based on how that eating disorder is affecting their body. Others might be better suited to outpatient care, and everybody's different. So I think having a good team, starting with family and friend support, including a therapist, maybe a nutritionist, and a psychiatrist that the patient feels comfortable and validated and supported by, within the broader context of a support network of other people who might be able to validate the experience. Some of this you can find online, if not in your home region.
Excellent advice, Rachel. Thank you. So I want to thank you for an excellent presentation, and I want to thank everybody who joined us today as well. I'm very pleased to share that BBRF has received a $1 million challenge match for 2023 from two very generous family foundations that are passionate about our mission. The goal of the match is to accelerate the momentum in brain research as further breakthroughs are still needed. You can participate in this initiative if you're a new BBRF donor, a past donor who has lapsed but makes a donation this year, or a current donor who increases your 2023 gift with your increased amount to be the amount that is matched. I hope you'll consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. We're able to do this because our operating expenses are covered by separate foundation grants. This means that when you donate $1 for research, that dollar goes directly to the scientists. Please make a gift by visiting bbrfoundation.org or call us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion of the presentation or would like to share it, please visit the events and webinar page on our website. Please join us again next month on April 11th at 2 p.m. Eastern Time when Dr. Ryan Van Lieshout, Associate Professor in the Department of Psychiatry and Behavioral Neurosciences, the Albert Einstein / Irving Zucker Chair in Neuroscience and Canada Research Chair in Perinatal Mental Health at McMaster University, will present Changes in Infant Emotion Regulation Following Maternal Treatment for Postpartum Depression. Thank you, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Take care.